Everybody, and thank you again to the DREAM organizers for holding this challenge. It's been a lot of fun to participate, both this year and last year. And actually, listening to the questions just now at the end, I think I'll be able to. This talk will hopefully address several of them, and if not, please ask again at the end. So I'm Po Ru. I'm the first member of this Orange Balls team, and I am finishing my PhD at MIT with Bonnie Berger. So Julio and Pablo already gave you more or less the picture of what's going on here. But just to recap very quickly, this challenge had 4 components, I would say. The first was they gave us a circuit diagram, a complete circuit diagram of all of the proteins and mRNAs in a network, and all the activation and regulation repression links. They also gave us the complete picture of the ODEs that govern the time evolution of all the species in the system. And that's actually very powerful, because that means that although we were left with 45 parameters to estimate, we know the exact functional form of the equations that govern the time evolution of all of these species. So there are just 45 constants that we need to estimate. So that was the setup, and then it gave us a budget and a cost schedule of what perturbations we were allowed to apply to this network and what we could measure. And finally, we were given the opportunity to purchase data which had this 20% Gaussian noise added to it. But importantly, we were told that we were getting data with 20% Gaussian noise added to it. And as you'll see, that was actually very important and very useful for our modeling. So that's the setup. We have these 4 sources of information, and we were asked to estimate the parameters. So given what I've just said, you might think, and correctly think, well, isn't this just a problem to apply maximum likelihood to? Because you've told me data, you've told me experimental measurements, you've told me a known noise model that the data that I'm getting has 20% Gaussian noise added to it. And therefore, well, why don't I just find the choice of parameters that maximizes the likelihood? Well, by and large, that's actually correct. That's more or less what you should do. There are a few small subtleties, namely that they clipped negative values to 0. There were some cases where the data after adding noise would fall below 0. So those got clipped to 0, and as a result, the likelihood function isn't exactly a Gaussian. But that's by and large not a huge deal. And in fact, maximum likelihood, I would say, is the right way to go. Except what makes this interesting, and what makes maximum likelihood not the end of the story, is this fact that there's this limited budget, and you don't just get a flat table of data that you're told go analyze. You have this choice to make. In fact, you have many choices to make of which data do you think are the most useful, and how do you go about choosing which data are the most useful, especially at the beginning when you don't know a whole lot about the system to begin with. In fact, because there were 2 choices to make in any data purchase, namely what perturbation do you apply and then what do you measure. That made for a combinatorial number of possible experimental combinations. So there are probably a couple hundred different possible experiments you could buy, and as Pablo said, you only have money for maybe 10 or 20 of them. So an analogy that I'd like to draw to how we approach this problem is to think of it as a game, as a one-player game, in which we're iteratively playing this game by making measurements, using up some of our budget, and then looking at the data that we got and iteratively deciding, okay, based on the data that we just got, what do we do next? So this is in game theory terminology, this is a one-player game with a finite number of moves, again restricted by the budget, and we get some hidden information that's revealed after every move. So clearly, optimal play would involve reevaluating the strategy with the new information that you get after every move. And so you get this kind of branching game tree, abstractly at least. One thing that I want to mention is that an initial idea that you might have as a first approach would be to do basically a greedy algorithm, whereby what you do is you have some notion at any given time, based on the data that you've collected so far, of how well constrained each of your parameters are. And given this sort of uncertainty space that you have on your parameters, you could then think, well, let me just identify the parameter that I know the least about, and let's do the experiment that will reduce the uncertainty as much as possible. And that's not an unreasonable idea, but I think what we realized that ultimately led us to develop methods that set us apart is that it's not enough really to just look at one parameter at a time. It's kind of clean mathematically to think about it that way. But in reality, if you just look at one parameter at a time, or if you just think about one move at a time, one purchase at a time, you lose a lot of information about what you actually know about the problem space. So in this cartoon, what I've shown you is let's say with two parameters, P1 and P2, you could imagine your uncertainty space being, well, sort of a blob, that both P1 and P2 are relatively unknown. But you could also imagine your uncertainty space looking sort of thin and narrow, but oriented in such a way that you don't really know too much about P1, and you also don't really know too much about P2, but you do know that they are either both high or both low. And that's useful information that you should take into account when you're deciding what choices of data are going to be most useful. So I just said it's important not to restrict yourself to thinking one move at a time. And in fact, you could think about going about this from the completely opposite end, which would be, well, ideally, if I had all of the computational resources and all of the time in the world, I should just think about all possible game trees. And just, since there's a finite number of budget-limited moves, just write them all out and put some prior on what I think the parameters are. And then for every tree, calculate the expected value that I get out of following this game tree. Do this over all the trees, minimize the expected error, and there's my strategy. So this is also not a good idea, but for the opposite reason that it's just impossible to do computationally. If you could do it computationally, it would be a great idea. So then the question is, what can we actually do that's computationally feasible? And so I want to use an analogy that I brought up last year, and I still kind of like, which is an analogy to chess. So chess is another kind of game where calculating a game tree in full is completely intractable, and that's kind of clear. Again, at any given stage in the game, you have probably 100 different moves that you could choose from, and even looking a few moves ahead will give you an exponential explosion in the number of game states that you have to analyze. Nonetheless, we all know that the folks at IBM did a pretty good job on Deep Blue. And that chess computer typically looked something like 6 to 8 moves ahead. Apparently, sometimes it even looked up to 20 or more moves ahead. And so then the question is, how did it do that? And there are really two main ideas that were used in Deep Blue, and which actually apply pretty well to the way that we approach this problem as well. The first being that it had a very good evaluation function of being able to estimate how high quality is a given move without having to follow it down many steps and see what a potential outcome would be at the end of the game, because you simply can't calculate that far. The second was using heuristics that would basically allow you to prune your search space and massively reduce the combinatorial explosion of search space that you have to go through. One thing that I should mention that someone last year got slightly confused by this analogy, because, well, the thing is, chess is a 2-player game. What I'm playing in this challenge is a 1-player game. Also, it's not an adversarial game. The dream organizers aren't going and changing the parameters every time I buy something, or at least I don't think they are. So there's some slight differences, but overall, I would like to think of this as a game, and a game where you're getting some small bits of new information, having to reevaluate your strategy as you play. So back to those two key ideas. again, the same two key ideas are important in the way that we play this particular parameter estimation game. And so then the question is, how do these translate to how we approach it? First, the idea of finding a good evaluation function. We found it very important to get a rough idea of how much information was available in any given data purchase. And also, how much information do we need to narrow down the uncertainty in a given parameter? Which of these parameters are easy to get? Which of these are hard to get? And having heuristics to understand that and get a handle on that was really important. Secondly, having heuristics to reduce the search space and to do pruning, to limit the amount of game tree options we needed to consider was also really important. And I'll talk a little bit about each of those. So overall, the way that I've cast our approach is sort of a hybrid between, originally I had these like 4 areas, and I showed this picture of, well, you collect all of your data, give it to a computer, and have it sort of crank through it. In fact, what we found to be highly useful was, well, let's look at these four aspects, think about it very hard, and give it to a computer afterwards. So now at this point, I'm just going to go through a little bit of math to give you a sense of what solving this challenge actually amounted to in practice, and in particular, how these equations behave and how the data behaves that we're given. So what I've shown you in this slide is just the most simple time evolution equation that occurs in the system. And this is just a single mRNA that is not being regulated by anything else. And therefore, just given by the time derivative of mRNA is equal to some rate at which it's produced minus some degradation rate times the concentration, times its own concentration. So if you write this out in variables, you can see very rapidly, actually, that this time evolution equation is given by just a simple exponential. It just reaches a steady state and rises according to an exponential to a steady state. So this is very useful and very important for us in our analysis because, well, here's an example of the data that we would actually get when we purchase. And this obviously doesn't look like an exponential. It looks like maybe an oscillation. But the fact is it's supposed to be an exponential. And the fact that we know that it's supposed to be an exponential means that we can essentially say, OK, it looks like there's an oscillation here, but we know it's not. These are actually just all a bunch of values that are supposed to be equal, and they've had 20% noise thrown onto them. But we can just use square root of n and get rid of that, reduce our noise, and estimate our parameter. With proteins, it's actually a very similar case still applies, which is that the protein concentrations, by and large, also go to a steady state. In this case, the steady state value that they go to is a little bit different. It typically looks slightly more complicated and is some algebraic combination of a few different parameters that we're interested in. But again, if we work this out on paper that this is what it's supposed to go to, then we know all of these values are actually supposed to be equal. We can average them out and get a good estimate for this product and ratio of parameters we're interested in. An additional slight subtlety is that we can actually get more information from this picture than just the steady state value. The transient value is actually also useful and lets us get a window into some other parameters. Finally, one last slide of math goes back to these activations and regulations, which add to the complexity of the system. And the nice thing, though, is that, again, we have explicit equations that tell us exactly how these work. And in fact, they obey these hill multipliers. The interesting thing here is that, which helped us to guide our choice of experiments, is that the hill terms only do interesting stuff over a narrow transition window around the KD, the binding coefficient. And as a result, it was really critical to try to target our perturbations so that it put protein concentrations within this window in which we would get information about the hill coefficients. So to summarize very briefly, steady state concentrations were extremely helpful for basically reading off a few types of values. And then as for getting the Hill coefficients, it was really important to put protein concentrations within a transition window. So that takes us through the first aspect. And then briefly, just on the second, about reducing the search space. We used two main heuristics for this. One was to just reveal information early on, whenever possible. That one was kind of obvious. If there are experiments that we know we have to do sooner or later, then let's do them sooner than later. And that way, we can get information that will inform our later purchases. And then secondly, importantly, this Pablo also alluded to earlier, which was the idea of isolating independent components. And going back to something I mentioned earlier about trying to analyze, to co-analyze parameters and to look more than one move at a time. This, I feel, was actually an extremely important part of our solution process, which was that if we can narrow down on just 2 or 3 proteins at a time, that greatly aids our ability to do this co-analysis and to analyze sort of in multiple dimensions without having things completely blow up. So just to wrap up now, choosing the actual purchases was sort of a hybrid process for us of kind of staring at data values, scribbling a lot on pieces of paper, and then also putting it into MATLAB and getting some computational checks on whether or not it looked like particular data purchases were going to help us. Typically, the first purchases were always the hardest. We would kind of stare at all of our initial startup data values for a long time before we made any decisions on what to buy, and then things got faster after that. Very briefly about the results that we got. The parameters that we predicted for Model 1 were usually good to about 10%, as you can see on this left chart. The time courses were actually accurate to about 2%, and that goes along again with the notion that there are some parameters that are poorly constrained, but the fact that they're poorly constrained is actually because they don't do much to the system. And so since that means that we don't estimate them very well, but the fact that we don't estimate them well doesn't have much effect on our predictions of the time courses. So finally, the key ideas, I would say, of our approach were this sort of game tree planning. And in choosing our data purchases, I like to call this a computer-aided human process, whereby we didn't have a codified algorithm that told us what to choose, but rather we did sort of back-of-the-envelope calculations, used a lot of simulation in the process, but ultimately made those decisions ourselves. As for the actual maximum likelihood fitting, that of course was done with primarily with just a black box optimizer with some intervention by us. And finally, as a future direction, an interesting challenge if you guys want to do it in future would I think be to expand the size of these models to the extent that you actually need to automate the whole thing. I think what I would claim is at this point for 6 to 10 proteins, a human-aided computer-aided human can probably beat a straight-up computer because there's this complex decision-making process to be done. On the other hand, if you gave me 24 proteins, I would throw up my hands and I'm not gonna crank through this all myself. So that's it. Thanks to the organizers again and to my teammates.
Thanks a lot. So we have time for questions.
Just a simple question. So one strategy could have been at each stage decide which is the most important parameter to look at according to sensitivity analysis. So go for that, circle that a little bit, and then move on to the next most sensitive and so on. So is that something that you considered?
Yeah. So that's something that I tried to sort of cast as a first approach. But what we actually found most useful was to think maybe 2 or 3 steps in advance and think about if we're going to spend, let's say, 2,000 credits on these 3 proteins, how do we want to optimize which 3 experiments we want to do? And we found that to be particularly useful.
Ernest?
Really great talk. I was wondering, since one of the points made was that knowing the functional form was a huge help.
Yes.
So do you think there's a way to modify the predictions
Yeah, I think the challenge there would be, when you set up a challenge, is that you want to not make the winner end up being the person who, has the best guess of what you thought might be, of sort of what you dreamt up. I think there are ways that you could do that, though. If you kind of, had 3 or 4 different possible functional forms or something and said, I'm going to choose one of these randomly, or have some parameter on the functional form.
Last question.
Yeah, hello. I wanted to know what are the key components of your heuristic to determine if a perturbation is interesting or not. Did you perform simulation, or is it just based on sensitivity analysis from equations?
So I would say the most important heuristic for us was the steady state analysis. So based on the steady state analysis, we could get a sense of if we hit a certain steady state, what parameters will we be able to pin down, and what combination of parameters will we be able to pin down. And based on that, then we could evaluate how useful is a given experiment to us. And I'm happy to talk more about that later.
Good. So thanks again, and